Have you ever wondered about the incredible process that allows your muscles to move, enabling everything from a simple blink to a powerful lift? This fundamental biological marvel underpins all our physical interactions with the world, from the smallest twitch to the most strenuous activity. Today, we'll delve into the fascinating molecular science behind muscle contraction, exploring the intricate cellular machinery and biochemical reactions that make movement possible. This process is truly a marvel of biological engineering, occurring constantly within our bodies. It all begins with a precise electrical signal from your brain, traveling rapidly down a motor neuron to reach a specific muscle fiber. This initial command is the fundamental first step in any voluntary movement we make. This journey culminates at the neuromuscular junction, a specialized synapse where the nerve meets the muscle fiber. Here, the electrical signal is converted into a chemical message, bridging the gap between the nervous system and the muscular system. Acetylcholine is released and binds to receptors on the muscle cell, triggering an electrical signal or action potential. This signal then spreads rapidly across the entire surface of the muscle fiber, ensuring a coordinated response. This electrical impulse then dives deep into the muscle fiber through an intricate network of invaginations called T-tubules. These transverse tubules ensure the signal reaches every myofibril within the muscle cell, initiating the next critical step. At the very heart of muscle contraction lies the sarcomere, which is recognized as the fundamental contractile unit of muscle tissue. These highly organized, repeating units are responsible for the muscle's ability to generate force and shorten, forming the basis of all movement and physical activity. Within each sarcomere, we find two primary types of protein filaments arranged in a precise, overlapping pattern: the thicker myosin filaments and the thinner actin filaments. Their specific arrangement and intricate interaction are absolutely key to the muscle's ability to function and contract effectively. The widely accepted sliding filament theory provides the elegant explanation for how these protein filaments interact to shorten the muscle. It describes a process where the filaments themselves do not shorten, but rather slide past one another, causing the overall sarcomere to contract and generate force. Myosin heads, which resemble sculpted marble tools, extend outwards from the thick myosin filaments and are absolutely crucial for initiating and executing this contractile process. These heads act as molecular motors, driving the movement and generating tension. However, in a relaxed state, the critical binding sites located on the actin filaments are physically blocked by another regulatory protein called tropomyosin. This blockage prevents the myosin heads from attaching prematurely, ensuring the muscle remains relaxed until properly stimulated by a nerve impulse. The electrical signal from the nerve triggers the sarcoplasmic reticulum, a specialized organelle, to release a flood of calcium ions. These calcium ions then bind to troponin, a protein associated with actin. This binding causes troponin to change shape, pulling tropomyosin away from the actin binding sites, exposing them. With the binding sites exposed, myosin heads can now attach to actin, forming cross bridges. Once attached, the myosin head performs a power stroke, pivoting and pulling the actin filament towards the center of the sarcomere. This pulling action shortens the sarcomere, and the cumulative shortening of many sarcomeres leads to muscle contraction. But how do the myosin heads detach and prepare for another stroke? This is where ADP, our body's energy currency, comes in. An ADP molecule binds to the myosin head, causing it to detach from the actin filament. The ADP is then hydrolyzed into ADP and inorganic phosphate, releasing energy. This energy cocks the myosin head back into its high-energy position, ready to bind to actin again if calcium is still present. This cycle of attachment, power stroke, detachment, and recocking continues as long as calcium and ADP are available. When the nerve signal stops, the muscle begins its journey back to a relaxed state. This cessation of stimulation is crucial. Calcium ions are actively pumped back into the sarcoplasmic reticulum, requiring ADP. This energy-dependent process reduces calcium levels. As calcium levels drop, troponin and tropomyosin return to their original positions, blocking the actin binding sites once more. Without available binding sites, myosin heads can no longer attach to actin, and the muscle fiber lengthens and relaxes. This intricate dance of proteins, ions, and energy is a testament to the elegance and efficiency of our biological systems. From the smallest twitch to the most powerful contraction, every movement is orchestrated by this precise molecular mechanism. Understanding this process not only deepens our appreciation for the human body, but also informs fields from medicine to sports science. Thank you for joining us on this journey into the science of muscle contraction. We hope you gained a deeper understanding.